This is a P20, means I can take up to 20 microliter using this pipette, okay? And now I can set 12 microliter. 12 microliter. Of water. I'm taking only water first, so I can use the same tips for all the four tube we have, separate tubes, because it's empty and sterile. There is nothing in it. Okay. Now, but from next edition, I cannot use the same tips for all these tubes. Yeah, then it will be cross contamination.
So is that deionized water?
Yeah,
Yeah.
Sterilized, deionized water.
Okay.
We have autoclaved it.
Cool.
Okay, and so whenever you add, you just put a tick mark. And I've already added water, and I'll just take PSA 1 microliter.
So how did you decide it was going to be, what the volume of the reaction should be? Like,
I'm sorry.
so how did you?
How do I know whether what should be the total volume?
Yeah.
Usually, it's we like to make it 50 microliter total volume for the restriction.
Okay. Right.
You can do it 25 microliter or whatever you like.
Yeah.
But I took 50 microliter because like then I will have, hopefully I will have enough of vector digested that I'll be able to see on the gel. I need to run the gel after the digestion, and then I'll be able to see in the agarose gel hopefully 2 separate bands for your parent vectors and 1 thick band for our expression vector. Yeah.
So what is this one?
This one is BSC.
Okay.
This one is BSC.
So what is the BS?
Actually, I'm sorry guys, because it will be quite impossible for me to remember all your names at this point, but yeah, that's why I cannot just call by your name at this point. Yeah, and we also made name text ready for them.
Okay.
Anyway, this is the first time.
Sure.
Hopefully.
Do you have any labels that I can write names on?
Maybe whatever you guys can do, we'll have the masking tape. Yeah. Yeah. Name and just stick it here or something maybe. Okay. Yeah. So I have added already. Okay, so this buffer is actually 10 times buffer, and our total volume is 50 microliters. So how much should I take from here? Any idea? This is a 10 times buffer, and I'm gonna take this buffer for a total volume of 50 microliter. So how much volume should I take from here?
Slowly down.
5. Okay, I tell you why. So this is 10 times. So if I take 5 microliter, it is up to 5 times 10. That means 50 microliter. For 50 microliter, it's enough. So as long my total volume is 50, I'll take only 5 microliter from here. Is it clear to you guys? Any confusion? At this stage, hopefully not. 5 microliter or 10 times buffer. Do you guys know each other before or
Kind
no?
of. We've known each other for several hours.
Oh, okay.
That's true. That's all you really need.
That's what I'm saying.
Your hand right. Your hand.
Oh yeah, I'm feeling good. Can somebody write down like all the materials we're using?
I'm just gonna list it.
Oh, can I?
The amount.
Oh, okay, no worries.
Yeah.
All right.
Correctly
Not
zoom in on
point.
that.
So it's a good practice to use the enzyme very last. And also I don't have my enzyme here, it's in -20. So once I'm ready, I have added everything, then I'll just take this enzyme out and add it. Okay? Yeah. So I have only one more thing left, that's my vector. I'm gonna take 30 microliter each. So this is our MAC 1 plasmid. PMK MAC 1. This is our RGD plasmid. PMK RGD. This one is B221. Here. Okay, I'll just get the enzyme. Good trick is to get your ID bucket with you and get the enzyme we're using here. Okay? Not from New England Biolab. Okay? This enzyme. And I need 2 microliter each. I have just enough.
That's good.
I was scared, like seeing that. Oh my god.
We'll have to buy more of that, obviously.
Okay, that enzyme.
Now, do you need to use a new pipette tip for every one?
Sorry?
Do you need to use a new pipette tip for every?
Everyone, yeah. Because now I have to use fresh pipette tips, right? Any idea why? Yes, otherwise they will all be mixed up, right? So this point I have to use fresh pipette tips each and every time. Otherwise I will contaminate my enzyme as well as I will like cross-contaminate all my stuff.
Ash,
Yes.
if you have full vial of restriction enzyme, would you
Yeah.
aliquot it out for your experiment?
Yeah, you can do
You
that.
normally
yeah.
do that?
Yeah.
It's safer that way.
Yeah. Yeah. Oh my god. I don't have. Shit. I need to give it a spin because I cannot take out through my printer from here. Which one is that PMK mag?
Okay.
More protein.
Sean, sorry to say.
Don't like to hear that.
Not enough. Okay. So what I like to do at this point, I'll just transfer all my stuff from here to that vial. So hopefully in that way I'll get the maximum of my enzyme.
Should the drops count? Yeah.
Yeah.
Decide.
Micro drops.
Yeah, it adds up. No.
So before that I like to mix. Eventually we have to mix them. Do you guys get my point? Like what and why am I doing this? Because we don't have enough restriction enzyme at this point. I just realized. I thought it would be enough, but just not enough. Okay. So now this tube is our PMK mag 1. Okay? Yeah. So we can do that. We can flip and then spin and then allow them to digest at 37 degrees Celsius. So enable it. Mag 1 PMK mag 1. For the mixing, what you can do this sort of flips, or you can use your pipette tips, just pipette it up and down. Okay. So as long it's not a huge volume, big volume, that's why I'm just flipping, because whenever you take another fresh pipette tips and do the pipetting up and down, eventually you will lose some. You know what I mean? It will just stick to the outside of your pipette tips sometimes. You can do that, but I think that's okay. Now we'll just spin them. I don't need this. I'll just spin and then leave them. Okay, what you can do, you can just put them into the heat block and set the temperature at 37. Otherwise, what I usually do, I don't follow. I usually put them into the 37 incubator. Just let them digest right there. The whole stuff, just put them inside the calendar, And actually, it's not a bad idea, because then everything is in at 37, not only just the bottom part of your tubes.
So, I suppose we could go over there.
So for the centrifuge, do we need to change the settings or just as it is, we just put in the equipment?
It's a good question. So, yeah, we should ask.
Whenever you do the spin, make sure it's balanced. Like we have 4 tubes, so it's easy. I'll just give it a pulse.
So,
Yeah.
what settings are you using? This one. I guess you're just using the pulse setting, isn't it, right?
Yeah. So
Yeah.
Now I'm giving at high speed, maybe next couple of seconds. Just go for maybe 25 seconds.
You can also like program it to do like 2 minutes or something, but yeah.
Now we need to see whether there is any bubble. It looks like no bubble, right? Well settled at the bottom, right? So now we can allow them to digest.
So why did we need to spin it?
Yeah, just to settle them at the bottom, and
Oh,
yeah,
right, yep.
without any bubbles.
Okay.
Yeah.
Yeah.
Yep. Now we'll just put them into the incubator. Let us say for next maybe 1 hour, 1 hour 15 minutes. So we have the incubator here. So this is at 37 degrees Celsius. Yesterday we did the transformation for PMK RGD and PMK MAC 1 as well as PLP1. So yesterday evening we did a transformation and shown these are the plates. It's growing, but you see the colony are really tiny.
Yeah.
Because not enough plant. It was just overnight. So at I think afternoon time you will see their colonies will be quite big. Can you guys see small colonies? It will be bigger later this afternoon. So we have to work with this one as well. Oh, I need to take this one, the BL21 and grow.
Okay.
You know, 4 mil. Yeah, I'll just take here and I'll just leave this one.
Kind of. I think this is separate from what we're doing now, isn't it?
Yeah.
Yeah.
I'll explain later. So
okay, don't worry.
About
Yeah.
This one. We just incubated juice.
Okay. Going in well, or everything makes sense? That's good.
If you guys see this sheet, actually we have divided our work into